Welcome to viruses. Possibly a little strange saying welcome to viruses since we've been living with a virus for the last while and all the constraints of that virus. From a biological viewpoint and from our course viewpoint, viruses are fairly straightforward. Quick enough chapter, not that much that you need to know, and they're quite an interesting type of living or non-living thing. So here are the learning outcomes, the things that you need to know about viruses by the end of this chapter. First of all, we need to look at the problem of defining viruses as either living or non-living. They have some characteristics of both, and we'll need to be able to list some of those characteristics and discuss why it's a problem coming down on one side or the other, saying that they are living things or they're not. Then you need to know that there's a variety of shapes of viruses. You need to be able to list those shapes. You need to be able to outline the basic structure of viruses, which is far easier than the basic structure of any of the other things that we looked at in the 5 kingdoms of life earlier in our 5th year course. We need to be able to explain the process of viral replication. Now, replication is when viruses make copies of themselves. Then we need to finally be able to look at the economic importance and the medical importance of viruses to humans, plants and animals. And you need to know at least 2 harmful effects of viruses, and there's some debate as to whether it's 1 or 2 beneficial examples of viruses, so no harm to know 2. So let's start with what a virus looks like. So you notice I'm not saying virus cells, or at least I'm trying very hard not to say virus cells, because viruses are non-cellular. They're not made up of cells. They have a much more basic structure, and they're tiny. They're actually 50 times smaller even than a bacterium. If you notice this bacterium here, a rod-shaped bacterium with a little flagella coming out of the back of it, you'll see that we have a teeny tiny virus down here, and it's actually much, much smaller than the bacterium. They're so tiny, in fact, that they can't be seen even with a light microscope. You'll need an electron microscope to see them. So let's get into a little bit more depth. No need to worry too much about the diagram up at the top of this slide, because the structure of your virus is actually really straightforward. A virus has 2 parts. It has a protein coat on the outside that's called a capsid, and then inside that it has a strand of either DNA or RNA. It doesn't have both nucleic acids. Higher organisms would tend to have both nucleic acids, both DNA and RNA. And if you're wondering about DNA and RNA, we'll actually be doing a full chapter on those next year in 6th year. But viruses will only have one or the other, either DNA or RNA, which is one way in which they are different from lots of living things. And if they only have that little one strand of DNA or RNA, you can imagine that they have very few genes. In fact, all they have really is the instructions for making new viruses, and that's it. Now, viruses don't have organelles. And if you remember all the way back to the start of 5th year, you'll remember that cells are made of organelles. So that should help to reinforce the point that these are not cells. They've no mitochondria, they've no ribosomes. We don't even refer to them as prokaryotic, because prokaryotic is a description for a cell. If you remember, we came across that when we were looking at bacteria. So viruses have no cell organelles. We don't even call them cells. If they don't have cell organelles, they can't carry out any of the processes of metabolism, any of those chemical reactions that go on in our bodies, for instance. They can't do them on their own. So they need the organelles and the enzymes of a host in order to do them. For that reason, they're referred to as obligate parasites. That'll be coming up again a little bit later. So now we've briefly dealt with the structure of your virus. It has an outer protein coat called a capsid, and it has the DNA on the inside. Now to the next question. Are viruses living or dead? You may have a very quick answer based on the fact that we did the 5 kingdoms of life at the start of the year and viruses didn't feature there. So they're dead, right? except it's not quite as straightforward as that. They're kind of on the border between living and dead. And you could be asked in your exam to come up with 2 to 3 characteristics that are living and 2 to 3 characteristics that are non-living. Or a common question is outline the difficulty of classifying viruses as living or dead. And what they mean by that is you state that it is difficult to classify viruses as either living or dead because they have some characteristics that are living and some characteristics of non-living things. And then you need to list a couple under each heading. So what are the characteristics that they share with the living things or organisms that we saw when we studied the 5 kingdoms of life? Well, they do have some genetic material. They have either DNA or RNA, like all of the organisms that we studied. They have a protein coat, which again is common to all of those organisms. And they can replicate, they can make copies of themselves, albeit with a little proviso here, or a little bit in brackets saying they can only do that inside another living cell. Right. If we contrast that with the non-living features then, and the layout of this table, which is taken from your book, isn't exactly the best, because really the non-living ones kind of match up some of them with the living ones. For instance, where we've said a living feature is that they contain genetic material, then a feature that's non-living is they only have one of those nucleic acids, either DNA or RNA. Living things have both. They do have a protein coat, but they're not actually made up of cells. They can replicate, we said, as a living feature, but guess what? They can't do it by themselves. So if they can't reproduce by themselves, then they're non-living, because if you remember, all of those living things had to satisfy the 5 characteristics of life. And one of those was they need to be made up of cells, and another one was they need to be able to reproduce. And then the final one here that doesn't match up per se with the 3 living characteristics, and that is they don't have ribosomes, they don't have mitochondria, and they don't have other cell organelles. So again, we have living features and we have non-living features. You need to be able to list at least 2 under each heading. So viruses have features of both living and non-living material. Once inside another living cell called the host, they can replicate, which means make copies of themselves with the help of the host cell. And so, if you like, they're displaying some of the characteristics of being alive. But because they can only do that inside another living cell, they are what's referred to as obligate parasites. They cannot survive and reproduce outside of another living cell, or at least not for long. As they're parasites, then they cause many diseases in humans, in animals, and in plants as well. And we'll come back to those at the end of the chapter. Now, when we're trying to study viruses, one of the difficulties is that we can't actually grow them on agar because, as we've mentioned earlier, they're obligate parasites. They can only be grown in living things. Also, since they don't have all the different parts of a cell, it's really hard to design a medicine to get rid of them, or an antibiotic certainly won't work against them because there's no cell machinery or mechanics, if you like, for the antibiotic to interfere with, because the virus actually uses its host's machinery inside its host cells. Now, no need to worry too much about those last 2 slides. The main one in terms of are viruses living or dead is that table. So if you could learn that one, you'd be fine. Moving on then to the shapes of viruses. Viruses are classified according to their shape, and they can be round or rod-shaped, or they can be what's called complex here over at the end. That's a complex bacterium. If you like, you can think of it as a more complicated looking shape. Some examples then of some viruses, and the scale we're talking about here is each of these are smaller than even 1 micrometer, which is thousandth of a millimetre. There's your cowpox virus, the virus that causes rabies, mumps, another round one. The flu virus there, another round one. The tobacco mosaic virus is a rod-shaped one. And a bacteriophage, which is a virus that actually attacks bacteria, which can be a handy thing to have, is a complex shape. And we've a lot of study done of that bacteriophage. Sorry, I skipped over the herpes virus is another round virus. So you need to be able to list those three shapes and give an example for each. So a round, a rod-shaped, and a complex. So easiest one, the flu is your round, rod-shaped is your tobacco mosaic virus, and a complex one is your bacteriophage. Quick learning check here. You might want to see if you can answer those questions. Now we're going to move on to look at what should be reproduction of viruses, but it's not, because viruses don't reproduce. Viruses are not capable of reproducing independently. They only replicate, which means they make copies of themselves, and they do that inside a live host. Here's a bacteriophage over here. You don't need to worry too much about all the different components of the bacteriophage. The main ones that we would be interested in here would be, if you remember them from our earlier diagram, that there's a protein coat on your bacteriophage, and inside that, in this particular one, we have some DNA. If you remember, we said that protein coat called the capsid will have either DNA or RNA inside it. So no need to worry about the other labels there. This bacteriophage, we're going to follow it through the process of replicating, which it can do every 30 minutes. So there are 5 stages in our replication: attachment, entry, synthesis, assembly, and release. Now, at first they sound quite daunting, but you need to visualize what's happening here. You have a virus attacking a bacterial cell. How is it going to attack the bacterial cell? How is it going to get in there and use that bacterial cell for making copies of itself? Well, first it's going to attach onto the bacterium, onto its coat. Then it's going to enter it. Well, in actual fact, it's just going to send in its DNA, so that's the entry. Synthesis is where it starts to make components of new viruses. So it's going to make up some new protein coats, and it's going to make up some new DNA. Assembly is where it puts those protein coats and those pieces of DNA together to make new viruses. And then finally, release. So that poor bacterial cell is going to burst and release all the new viruses. Now, we're going to explore that in a little bit more depth over the following slides. So, step 1 is attachment. So, your virus here consists of a nucleic acid, in this case DNA, enclosed in a protein coat. So, what is involved in attachment? The virus, as is indicated by the name attachment, the virus attaches to the host cell. So, this virus here is sticking onto or attaching to our bacterium. It does that by protein matching. So there are proteins on the virus that match up with corresponding proteins on the surface of the bacterium, but no need to go into that level of depth. So that's attachment. The virus attaches to the host cell, in this case, an unsuspecting bacteria. Stage 2 then is entry. And as the name suggests again, something is entering. So in this case, the viral DNA is being sent in to the bacterial cell. So it inserts its DNA or its nucleic acid into the bacterial cell. The protein coat doesn't actually go in, it just stays on the outside, because the DNA here has all the instructions that are needed to kind of hijack all the parts of the bacteria and use it to start making new viruses. Then on to synthesis, stage 3. Synthesize is to make. So the host DNA, first of all, is inactivated. So when the virus DNA comes in here, it deactivates the host DNA, and then it starts using all of the different parts of the host cell to make new nucleic acids and proteins. So it's making new DNA, it's making new proteins, it's making new parts that will eventually be put together to form new viruses. So that's stage 3, the making of these new parts, which is called synthesis. Stage 4 then is called assembly, and as you might be able to work out from the title, the DNA and the protein is put together to make a new virus. So put all of these little bits of DNA together with all the proteins, and you will have new viruses. And finally, stage 5. What happens in stage 5 is release. So the poor bacteria gives up the ghost at this stage, and it bursts. And the title for when a host cell bursts and releases all of the new viruses is called lysis. Lysis meaning splitting. So when that bacterial cell bursts, all of these new viruses that have been assembled in there will be released out. So that's step 5, release. Now that's fairly long-winded, the last 1, 2, 3, 4, 5 slides that I've done, but you can summarize it all in one slide. So here we have our first stage up at the top here, and that first stage is when your bacterium attaches. Okay, so this is our bacteria attachment. That's number 1. Okay, and then what happens? Sorry, your virus attaches to your bacteria here. That's attachment. Then your virus sends in its DNA, so that's your second step, which is called, can we remember? Entry. Okay, then we go on to step number 3. The DNA has now entered, it left the protein coat out on the outside, and that DNA deactivates your bacterial DNA, makes it pretty useless, and it starts synthesizing. So here's our next step. There is synthesizing. 3, synthesizing. Or that step is called synthesis. The rubber working around it. That'd be too easy. So it's synthesizing there, so that step is called synthesis. And then 4 and 5 inexplicably are shown on the same diagram. So here we have a new virus being assembled, so that's assembly. So you can see that it has been put together. And then we have our bursting and our release of our viruses. So that's step 5. Okay, so those are the 5 stages of your viral replication, meaning how a virus uses a host like a little unsuspecting bacteria there to make copies of itself. Right, so here's a little picture of that last phase there, the lysis. So you can see this bacterial cell here is bursting to release all of the viruses that have just been copied. Time for another learning check. Have a look at these questions and see if you think you can answer them. Now, the last section of this chapter involves looking at the medical and the economic importance of viruses. You definitely need to know 2 harmful examples and 1 beneficial example, or maybe it's 2 beneficial examples, as I mentioned earlier. People aren't entirely sure about that, so maybe best to learn 2. So disadvantages are easy to come up with, I would say. human diseases, first of all. So viruses are responsible for or measles, mumps, and rubella. Um, the normal cold that you would get is caused by a virus. Flus are caused by viruses. Warts are caused by viruses. The common warts that you could get anywhere in your skin are caused by a human papillomavirus, and there are many HPVs or human papilloma viruses, and one of those is responsible for genital warts as well, and another one is responsible for a lot of infections of the cervix, which would trigger cancer of the cervix, and that's why there is a HPV vaccine available at the moment for teenage girls to try to prevent cervical cancer by preventing the infections of the HPV virus. Then on cold here, you'll notice I have a little arrow to a lovely furry looking thing, which is our coronavirus, that of course we're doing a lot of talking about at the moment. So what is the coronavirus? Well, first of all, we shouldn't call it the coronavirus because coronaviruses are a group of viruses. They're responsible for fairly innocuous things like colds, not responsible for the flu. That's a different virus. But they've also been responsible for some other fairly major health events like SARS. You may or may not have heard of severe acute respiratory syndrome. It would have surfaced in the early noughties, I think 2002. It was gone by 2004 and was looking like a real global health emergency. Now, I think we only had one confirmed death in Ireland due to SARS, in spite of the fact that the death rate amongst those who were diagnosed with it was about 11%, which is higher than what we're dealing with at the moment with COVID-19. SARS originated, or is believed to have originated in China. And then MERS, which stands for Middle Eastern Respiratory Syndrome, that would have been, I think it originated in Saudi Arabia in about 2012, and it is caused by a different coronavirus. Now, the death rate would have been huge for MERS. I think it was somewhere in the region of 30 to 40% of people who got MERS died from it. There were no confirmed cases in Ireland, though, for MERS. Now, when we look at SARS there, the one that we have at the moment, our coronavirus, is actually relatively similar to the one that caused that severe acute respiratory syndrome. That was caused by SARS coronavirus 1, and the virus that we have at the moment is referred to as SARS coronavirus 2. So it's the second of those SARS type viruses because it brings on acute respiratory symptoms, which means very difficult breathing and major lung problems. Now that's the name of the virus then, SARS coronavirus 2. Although we're just calling it the coronavirus. And the disease that it causes then is called coronavirus disease 2019, or if you shorten that, take the CO for corona, the VI for virus, and the D for disease, you've got your COVID-19. So COVID-19 is the disease. Probably less important than breaking down all the different words and explaining their relationship to each other is to look at the symptoms and the prevention. So the symptoms include a cough, shortness of breath, high temperature, or called a fever. And if you have any of those symptoms, hopefully in the next couple of weeks, if you've even one of those symptoms, you will be encouraged to get tested straight away for the coronavirus. Now, how do you prevent yourself from getting it? Well, the fairly strong message is to stay at home. Of course, we can't stay at home forever, but right now that's the message that's there. Stay at home, stay away from everybody. And as we enter the period of easing of the lockdown and easing of the restrictions, we need to go back to the initial messages that were being given to us before we shut down our schools. And those messages were wash your hands, wash your hands frequently. A thing called respiratory etiquette. You'll see the diagram of respiratory etiquette here, which is basically if you are going to cough, cough into your elbow. Avoid contact. That's the hardest one, I think, at present. So you have to avoid shaking hands with people, avoid hugs, avoid hugging grandparents is a toughie, and clean. So clean everywhere, all surfaces that you come into contact with regularly. It is recommended that you clean them down with some form of detergent like a soap and water regularly, or even with a disinfectant if you have it, but soap and water is just as effective. So that's your coronavirus. Of course, the coronavirus, when we compare it to SARS or MERS, the death rate at the moment, while it seems huge and it is all over the world, the death rate is still down around the 3 to 4%, which actually makes it significantly less than either SARS or MERS. Now, I would recommend that if you're looking for information on our COVID-19, the disease, or on the coronavirus that causes it, you go to reliable sources such as the HSE website or the Health Protection and Surveillance Centre, the hpsc.ie, and you'll get reliable, up-to-date information that's not sensationalist, I suppose is the best way to put it, about coronavirus and COVID-19. And certainly you'll find that on the HSE website and on the HPSC website, they do attempt at least to present facts without any major interpretation or slant on them. So let's get back to our disadvantages of viruses. Certainly the coronavirus is a major one, but there are others. So what about plant diseases? Viruses can gain entry into plants by vectors or carriers. If you are doing agricultural science, you'll have recently learned about aphids or greenfly. Greenflies are a major vector for a lot of different viruses, and so you would always try to eliminate them from, for instance, your potato crop. And one of those viruses would be the mosaic disease. And you'll see here we have mosaic disease on a tobacco plant. It would also be found on tomato plants and on potatoes, as I mentioned there a while ago. And that's because your tobacco and your potato and your tomato are all part of the same family of plants, the Solanaceae, so they're susceptible to similar diseases. So that's one example of a disadvantage of viruses causing plant diseases. They also cause animal diseases like foot and mouth. You won't remember foot and mouth, but it was a pretty major deal out around the Cooley Peninsula before you were born. SARS, as I mentioned earlier, now I mentioned SARS in terms of humans, but SARS is what's called a zoonose as well, which means that it can be passed from animals to humans. And so is rabies, which is another animal disease caused by viruses. Now, there are a couple of advantages of viruses. One of those is that they can be used in genetic engineering. They have a very simple genetic makeup, and that makes them quite suitable for actually putting genes into other organisms. So they can be used to effectively inject a gene into a target cell, um, using the same process as they would use just to inject their own DNA. So if you can put your gene into their DNA, then when they go through replication and when they enter the cell, they will stick in that target gene as well. There'll be more on that when we do our study of genetics. Also, sometimes the infection, if you like, the virus infection in your plant will give a nice new variety of plant. Like you see that stripy pattern on the tulip there is actually caused by mosaic. They can also be used, if you remember back to our bacteriophages, to control infections, because if they can kill the bacteria, if they can make bacteria burst in their replication process, then they're getting rid of bacteria, and they may help us to reduce infections by our antibiotic resistant bacteria, such as our MRSA, our methicillin resistant Staphylococcus aureus. Now, let's do another little learning check before we do the very last bit of this. 3 disadvantages of viruses and 2 advantages, and explain the term vector as it is applied to disease. Okay, our virus chapter goes into a little bit of detail about AIDS, which is acquired immune deficiency syndrome. The cause of AIDS, it's a disorder in which a person can't make antibodies, and that's because they've been infected by a virus called the human immunodeficiency virus, or HIV. The transmission, HIV can get into the body through any body fluids such as blood or semen. Usually it would be transmitted by sexual intercourse or infected blood products or shared needles. It can be transmitted from mother to child across the placenta, but it's very, very rare, or it could be transmitted in breast milk. It's not contracted by touching, embracing, kissing, where there's not a major exchange of saliva, or by sharing utensils that might have a little bit of saliva on them either, or toilet seats. You need a relatively appreciable amount of an exchange of fluid. Okay, the effects then of our acquired immune deficiency syndrome, AIDS, the disease that you get after HIV enters your body. So first of all, when the HIV enters the body, it can stay dormant for a while. At that time, if you had a blood test, you would test positive for HIV, but you've not developed the disease yet, AIDS. The virus then starts to attack the white blood cells, and that means that the person can't produce antibodies and they can't fight off infections. So AIDS sufferers usually die from an opportunistic infection, just a normal infection that they would usually be able to fight off no problem, but because of their decreased immunity, they can't fight it off. It's estimated that about 34 million people suffer from AIDS worldwide, maybe somewhere in the region of about 6,000 people in Ireland. So how can it be controlled or prevented? Well, there isn't a cure at present, nor is there a vaccine, but there are a range of medications that have been showing promise in treating AIDS, and certainly in prolonging life of people who are suffering from AIDS. Prevention is the key still, and the main methods of prevention would be to avoid sexual intercourse altogether, or to confine it to one faithful partner, or use a condom. Don't use shared needles or toothbrushes or razors. Avoid contact with blood and bodily fluid from others, so wear gloves if you do have to handle blood or bodily fluid. And anybody with AIDS should not donate blood or semen or body organs. And that brings us to the end of our section on viruses. Um, just one thing that I didn't mention there would be the use of antiviral drugs. There are some drugs that work against certain viruses, like your Sovirax, you may have heard, which works against the herpes simplex virus, which is the one that gives you cold sores. And there's a growing range of those antiviral drugs that are used to treat viral infections, and they stop the viruses without affecting the host, which is a very difficult thing to achieve. And lastly, don't ever take antibiotics for a viral illness such as colds or flus, and that is because your antibiotic will not work against them, and this misuse of antibiotics contributes to the development of some resistant strains of bacteria on which our antibiotics do not work anymore. So that's the end of the virus chapter.